back to the another video of pulsating biology. This is the lecture number 3 for the chapter in which we were covering the biological molecule. And in today's video, we are talking about the food test. We've already covered the rest of the topics in the previous videos. In the last video, we are required to cover the topic that is the food test. In food test, which is further divided into the four kinds of the test. It consists of the iodine test, the emulsion test, which is for the fats, and we have the burette test, which is for the protein, and then we have the Benedict's test. Now we're going to discuss each of them in detail. Let's first begin with the iodine test. All right. So the iodine test is basically also called as the starch test because they determine the presence of the starch in this test. That's why we call it as the iodine test. Let's suppose if we test any food item, could be a potato or chips or a sweet or even a biscuit. We test by crushing it. So the first step for the iodine food test is that we crush the item. Whatever item of food we are taking, it can be any item that can be biscuit or a sweet, we are going to crush it to release the content. To release the content of the item that whatever we are taking in it. Alright, so the first step is that we crush the item. What happens is that after the crushing, we need to identify either the iodine is present or in a test or not. So for that, we need to add the iodine in it. We add some drops of the iodine solution in it, and what happens is that if the color is changed, then the iodine is present, and if the color is not changed, then we say that there is an absence of iodine. And what the color appears is that the color appears to be brown. If it remains brown, then we say that there is no presence of starch. But if the color changes from brown to black blue, or you can say the blue black, then we say there is presence of starch. Then we say that the starch is present, and if the color remains same, there is no change in the color, that is the brown color, it remains the brown, then we say that the starch is absent after adding the iodine solution and crushing the item whatever we are taking for the food to release its content. The change of color basically helps us determine the food test very easily, that if there is a change in the color, then we say that the item is or the certain substance is present, and if there is no change of the food color, then we say that that is not present. So this is all you need to know about the iodine test. Whatever we do is that add an iodine solution in the crushed item, and if there is a change of a color into the blue black from the color brown, we say that the starch is present, and if there is no change in the color, then we say there is an absence of starch. Let's move on to test number 2. All right, the test number 2 is the burette test. The burette test is said to be burette test because it's for the identification of the presence and absence of a protein. What is different and what happens in this test is that we add sodium hydroxide and we add the copper sulfate. Then we identify the change of a color. But what is special in this test is that we say that this test is very clear solution. We do not use the term transparent or translucent. We use a very biological term that is present also, and it is mentioned in your syllabus to use this term, that is the clear solution. Please don't mark this in your exams that it is a transparent or a translucent solution. We call it a clear solution, which is the proper term that is mentioned in your syllabus also. So what happens in the burette test? We add sodium hydroxide first into the crushed item. Again, we are going to take any food content or any food item, and we are going to crush it, and then we are going to add sodium hydroxide. And the third item that we are going to add is the copper sulfate. The copper sulfate is blue in color. We have also seen the copper sulfate very commonly in the laboratory. That copper sulfate is blue in color. What happens is, if there is a change of the color, then we say that the protein is present. And what is the change of the color? Since the copper sulfate color is blue, and if it remains blue, then we say that there is no presence of protein. And the protein is not present. But if the color changes into purple, then we say the protein is present. This is a very easy trick to learn. PPP. protein present when it turns purple. Please do remember this, that if there is a no change in the color, then the protein is not present. But if there is a change in the color, then we say that the protein is present. Also, the color intensity tells us the amount of protein that is present in it. The amount of protein. It tells us about the amount of protein. The proper the color appears, the more amount of the protein is present in the solution. This is all you need to learn about the Buret test. We have covered two tests. Let's discuss the third test. The third test to be discussed is the ethanol emulsion test. The ethanol emulsion test is for the presence of lipids or fats that are dissolved in the ethanol. It tells us that in this test we are usually dissolving the fats and the lipids in the ethanol. We are using what we are using dissolving. It is all about the dissolution of fats and lipids into the ethanol. This usually does not occur except for the test. So what we do is that similarly in the last two sets we take a food sample, any food sample. It could be a biscuit, it could be a cheese, it could be any item you like. Take a food sample and then we crush it. Once we have crushed it, we add ethanol in it. Since ethanol is the most important item of this. Ethanol emulsion test. We add the ethanol, and the third item to be added is the water. After adding the water, what happens is that the lipids and the fats start dissolving in it. Once the water is added, it becomes start dissolving in it. Then the two kinds of the results can be expected. Either it can be a clear solution or it can be a emulsified solution. Now, what is an emulsified solution? Emulsified solution is a cloudy white colored appearing solution that tells us that there is presence of lipid. Whereas in the clear solution, we say that lipids or fats are not dissolved in this solution. This is how you get to know there's a no change of a color. Instead, there's appearance of two types of the solution, that is a clear solution and an emulsified solution, which is emulsified solution is a cloudy white kind of a solution that tells us the presence of a lipid. I'll take an example by the help of which it would be an easy for you to understand the ethanol emulsion test in more detail. For example, you have eaten a paratha and then you go and wash your hands. You might have noticed that if you wash your hands with the water just, then you feel some sort of greasiness in your hands. But unless you wash it with the soap, you still feel the greasiness. Once you've washed it properly with the soap, the greasiness is gone. Similar happens with this solution. When the fats and the lipids are dissolved in an ethanol, there's an emulsified solution that it becomes greasy, and that tells that there is a presence of lipids. Similarly, when your hands become greasy, we say that there is a presence of lipid in your hands unless you wash it with the soap. It's a very common example that helps students understand about the ethanol emulsion test. Let's talk about the fourth and the last step. All right. So the fourth and the last test is the Benedict's test, which is for the reducing sugar. All of the tests are for a certain purposes, and this one is for the reducing sugar. What you need to do in this test, that you need to take 2 centimeter of glucose solution. Once you take the 2 centimeter of the glucose solution, in the same quantity, you need to take the Benedict's test. You need to take the Benedict solution again in the similar quantity, that is 2 centimeter, which you have taken the glucose solution, and you have to heat this. You have to heat this at 800 to 880 degree to 100 degree temperature, which is the boiling point of water. And then you will observe that there is a color change. If the color changes from blue to brick red, we say that there is the reducing sugar present. But if the blue remains blue, then there is no presence of the sugar. No presence of sugar. By now, you might have you must have learned actually. By now, you must have learned that whenever the test in the test, whenever there is a change of a color, then we say that a substance is present, and whenever there is a no change of the color, we say that the substance is absent. However, this test is majorly also known as, we call it the semi-quantitative test. What is this? What is this called? It is called the semi-quantitative test. And why is it so? It is so because it goes through several stages. And by several stages, I mean that there is a difference of colors. We say that it first goes from blue, then it goes to green, then it goes to orange, and then at last it goes to brick red. At what color, what situation is taking place? Let's learn about it. In blue, when a solution remains blue, there is no presence of protein. All right. When it turns green, there is a tracing of reducing sugar. And there's no tracing of reducing sugar. There's not a large amount of a sugar present when a solution is green. When it turns orange, we say that there's a moderate condition of reducing sugar. But when it turns brick red, we say that there is a large amount of reducing sugar that is present in it. So this is the condition in which we say that the Benedict test is a semi-quantitative test where it goes through a different colors and then it reaches to a brick red where we say that there is a large amount of reducing sugar present. And this is all you need to learn about these four tests. And thank you for watching. The chapter we have completed the chapter. We've discussed about all the topics that were asked for us in the syllabus. So that's it. Thank you for watching.